Hey everyone, welcome to Professor Long's Lectures in Microbiology. I'm Professor Bob Long. As you know, these videos are intended to provide a basic and a fundamental understanding of some of the foundational principles in microbiology. By no means are these very detailed and extremely high-level explanations of some of the information that we could go over. If we do that, then sometimes we lose too many students. So my goal is to provide some of the fundamental concepts and a basic understanding, and then as you add more details, they actually fit in better rather than memorizing the encyclopedia. Now, please know that however your instructor wants you to learn the information, you need to learn it that way to prepare for the exams that you're going to face. My approach is to start very fundamental and basic, and then we will add details over time. Now, the series of videos we've been doing recently have been on genetics. So we were looking at the structure of DNA, how is DNA used to make RNA, messenger RNAs, as well as proteins, okay, protein synthesis. There were four major organic compounds inside the cell. We had carbohydrates, which we use for energy, although they can be used somewhat for structural things like adding into lipids like glycolipids, adding into proteins like glycoproteins and proteoglycans. But for the most part, our fundamental understanding is that carbohydrates are used for energy. They're big chunks of sugar. We break those down to release sugars like glucose, turn that into ATP. We've gone over metabolism in some detail. The second major class was lipids. Lipids are also carbon, hydrogen, and oxygen. They are used for cell membranes and organelle membranes, although prokaryotes don't have a lot of organelles. They have ribosomes, which are non-membranous, and that's about it. But for the cell membrane and the outer membrane of bacteria, those that have them, we do use phospholipids. So lipids can also be broken down for energy if necessary. We also have proteins. Proteins are long chains of amino acids, and they are used as the functional molecules of the cell. If some function is performed inside the cell, it's usually done by a protein, like enzymes that break down glucose into ATP and pyruvate. All of those proteins are performing functions. There's structural proteins, there's membrane proteins and transport proteins, as well as enzymes that perform biochemical functions. And then the last group was nucleic acids. Nucleic acids include DNA and RNA, deoxyribonucleic acid and ribonucleic acid. They can be the genome of organisms. Almost all organisms have the genome made of DNA, but there are some viruses that use RNA as their genome, and we'll talk about that in more detail. But the central dogma that we've discussed is that we go from DNA to messenger RNA to protein, and the protein performs the function. We've talked about the structure of DNA, how DNA is used for replication and the enzymes involved. We've talked about the expression of a gene, which we call transcription and translation. When we talk about gene expression, which is the topic of today's video, we're talking about going from DNA to messenger RNA to protein. We're expressing the gene as a protein. And remember, the genotype is the genetic makeup of the cell. What genes does it carry? Not all genes are expressed. Those genes that are expressed result in some kind of physical, observable characteristics of the cell. We call that the phenotype, the observable characteristics that are coded for by the genotype. So, we've done all of that stuff, and this is a brief review of that. What we want to talk about today is how do genes get expressed? Cells can have a single chromosome if they're a prokaryote, like a single circular chromosome, like bacteria, or they can have multiple linear chromosomes, like our cells and most other organisms. One chromosome, it's a long piece of DNA that contains many genes, and each gene is the instruction for the order of amino acids in a protein. That's the basic concept. And since the proteins perform the function of the cell, we say that DNA is the control molecule of the cell. It's the one that controls what's going to happen. And when we express a gene, that means we're going to transcribe that gene from DNA into messenger RNA, and then that goes to the ribosome and gets translated into a protein, and the protein performs a function. Again, not all genes are expressed. Something I want to get across to you, when we do this stuff, so let's say I have a molecule of DNA here, okay? I'm going to have a double-stranded DNA molecule. When I express it, we're going to open up the area where a gene is, and we're going to build a messenger RNA using an enzyme called RNA polymerase. When we're done, the messenger RNA comes off, and the DNA molecule gets put back together. Okay? Now the mRNA, also sometimes called the transcript, the process is called transcription. The mRNA then goes to the ribosome, and as the ribosome begins to read the messenger RNA, starts assembling proteins using molecules called tRNA. As the tRNAs bring in amino acids, they get bound together. The ribosome will zip down the messenger RNA, and by the time the ribosome gets down to the end of the messenger RNA, behind it, as it's holding the last couple of tRNAs here, it has all these amino acids bound together, and we've assembled a protein. When the ribosome comes off, I have a long chain of amino acids, and that protein will fold up. This is the primary structure of the protein, the order of amino acids, and then the protein will fold up into a tertiary structure and then perform some kind of function. We call that gene expression. This gene is being expressed in the form of some kind of protein or enzyme and a function in the cell. Now, as I stated a second ago, one chromosome can have hundreds, if not thousands, of genes. For example, when we look at E. coli, Escherichia coli, the bacterium that lives in our colon and our feces, E. coli has over 4,400 genes on its single circular chromosome. Those genes are not all expressed at the same time. And what I wanted to get across to you is protein synthesis, transcription and translation, or gene expression, is extremely expensive for the cell. It's very costly. It's costly in the form of energy, ATP. So the cell has to burn lots of energy. Just like you, when you have to burn a lot of energy to do something, you get worn out over time. And then you need to rest and replace the energy by consuming nutrients. Well, we don't want to burn energy if it's unnecessary. So if you mowed your lawn this morning, there's no need to go back and mow the same lawn tonight. Why would you mow your lawn every single day? It's a waste of energy. And it's a waste of supplies as well, the gasoline, weed eater line, and all sorts of other things. So when we talk about protein synthesis or even DNA replication, but protein synthesis in particular, it costs the cell a lot of ATP. The binding of the ribosome, the transfer RNAs binding in, the assembly of the amino acids, the making of the transcript, the rewriting DNA and the messenger RNA, all those chemical reactions and bonds very often require a lot of energy. So it's expensive for the cell. It's expensive for the cell in a second way, in the terms of resources. So if you ever watch any survival shows, you notice that people are always starving for protein. No matter how much, plants and other stuff they consume, at some point our body must have protein. To replace all the proteins that are breaking down and all the enzymes in our body. We're constantly recycling the proteins in our cells. And so in order to build proteins, we have to consume protein, break it down into the amino acids, send it to our cells, and then they use protein synthesis in order to reassemble those amino acids into human proteins. And bacteria would reassemble them into bacterial proteins. So in order to build protein, you have to have protein and amino acid. Now, most organisms don't know where their next meal is going to come from. So when you do have the opportunity to get enough protein, you don't want to waste those amino acids unless you absolutely need them. So why would I go assembling proteins that are not necessary? Not all proteins are necessary for the cell all the time. For example, if I were to look in a bacterium, we know that the bacterium can take glucose in, and then that glucose will be broken down by a process called glycolysis. And glycolysis uses a lot of proteins and enzymes to convert glucose into pyruvate plus 2 ATP, right? And this is the energy that we need. Well, the enzymes that are involved in glycolysis are needed all the time. So if I need a gene to perform a function, a protein to perform a function all the time, then that gene is pretty much always going to be on. And we're always making the enzymes for glycolysis. We're always replacing enzymes in the cell membrane, or not enzymes, but proteins in the cell membrane. Some proteins have to be made 24 hours a day, 7 days a week for the cell to perform its normal function. We call those proteins housekeeping proteins or housekeeping enzymes. They're doing the daily keeping up of the cell. So they're called housekeeping enzymes or housekeeping proteins. Their genes are on all the time, and we really don't ever turn them off. But some genes are only needed under special conditions. So when a cell encounters special conditions, it may need to produce a protein or an enzyme for that function for a limited time, and then no more. So if I don't, for example, our cells, for example, bacteria like E. coli can break down glucose. There's also another sugar out there called lactose that bacteria can break down for energy. But if glucose is highly abundant, I don't need to break down lactose. First of all, when I looked at lactose, lactose is what we call a disaccharide, where glucose is a monosaccharide. It's a single 6-carbon sugar. A disaccharide is 2 simple sugars in a chemical bond together. And it turns out that lactose is made up of a 6-carbon sugar called galactose bound to another 6-carbon sugar called glucose. And they both have the chemical formula C6H12O6. The order and the arrangements of the carbons and hydrogens and oxygens is slightly different. Now, there's an enzyme that can break this bond called lactose. It's actually called beta-galactosidase, but we're not going to get into that. Not lactose, lactase. ASE at the end of a word usually denotes that it's an enzyme, right? So lactase is a class of enzymes that can break down lactose. We're going to particularly look at one called beta-galactosidase, and you don't need to really know all that detail. If I run this molecule past lactase, I get a galactose plus a glucose as independent sugars. And each one of these can now be converted into energy. If free glucose, when we say free glucose, what I mean is glucose that's not bound to any other molecule. If free glucose is abundant, then bacteria prefer to use it. E. coli will prefer to use glucose for glycolysis to make its energy to survive. But if glucose becomes scarce and lactose is present, then the cell can turn on the gene to make this enzyme to break down lactose into glucose plus galactose and then convert those into energy. I don't need this enzyme, I don't need the gene for this enzyme unless those conditions are met. So if glucose is present, this gene is usually turned off. Or if lactose is absent, this gene is turned off. But the minute lactose becomes available, the cell will turn on the gene to make this enzyme to break down lactose into glucose plus galactose. And then it can use those for energy. And then if all the lactose is used up, then the gene can turn itself off again, and we go back to using glucose. So genes can be turned on and expressed through transcription and translation, and genes can be turned off so that we're not wasting the energy and the amino acids to make this enzyme, and we can save them for enzymes that we need all the time. Does that make sense? I hope so. So we're going to look at this process of turning genes on and off. And the thought is that there are many different ways that genes can be turned on and off within cells, and many of those ways have not been elucidated or worked out yet. But there's a few that have, and one of them is called an operon. And this is kind of the focus of today's lecture. So the big picture I want you to get from this is that gene expression involves transcription and translation, going from DNA to messenger RNA to protein to perform a function in the cell. The genes that code for those enzymes can be turned on, and if they're on all the time making enzymes, we call them housekeeping proteins. But some of them are only needed under special conditions. Because protein and synthesis is expensive in the terms of energy and resources, amino acids and other things, we don't want to be making proteins that we don't need. So the cell can turn certain genes off, and it will only turn that gene on and make that protein when it's needed, When it's no longer needed, it can turn that gene off and conserve the energy and the amino acids. And so this process is called an operon. Okay? And when we look at the definition of an operon is the series of genes under the control of an operator region. Okay? Now, some of the terminology I'm going to use is not going to be super clear until we see it sort of visually. And so, here's what happens. When we look at a series of bases on a DNA molecule, okay, so here's a piece of a chromosome. This is the circular chromosome of E. coli. There are a series of 3 genes here that are coded for that make the proteins that allow lactose to be entered into the cell and then to be broken down. And they are called the LAC-Z gene, the LAC-Y gene, and the LAC-A gene. And these 3 genes are necessary to make the proteins that get lactose transported into the cell and then get it broken down into energy. But I don't need these genes unless lactose is present. Well, there's also a section that always tells the RNA polymerase, hey, here's where we're going to start. Before we get to this gene, you're going to start here, and you're going to get ready to start reading these genes and get sort of in the right reading frame. Which bases do we start with? And that area of DNA that tells RNA polymerase where to start is called a promoter. A promoter is a series of bases that tells RNA polymerase, start here, and then start making the protein. And it gets it all lined up, because it can start reading 3 bases. If it starts with the wrong one, then it's going to read the bases out of order. We might get a different protein. So it sort of gets RNA polymerase lined up and reading properly and tells it, start here and make these 3 genes. Now, not all cells have this, but some cells have an area called the operator. And the operator basically controls whether the gene is functional or not. And there's another protein that's expressed by another gene that's a little bit uphill from this, or upstream from this. Since we're going to be reading in this direction, we call that downstream and this is upstream. That codes for a protein called a repressor. A repressor prevents transcription of these 3 genes. And the repressor will bind to the operator and somewhat overlap the promoter, so RNA polymerase can't bind. So here's the deal. We have a molecule called RNA polymerase, which is an enzyme, and RNA polymerase wants to bind here and start transcribing these genes. Well, and I'm going to make RNA polymerase smaller than this, but RNA polymerase, this molecule, wants to bind to the promoter and express these 3 genes. But in the meantime, there's another protein that binds here and blocks it, and that R is going to stand for the repressor protein. So the promoter is the region on the DNA that tells RNA polymerase bind here and start transcribing this gene. It's upstream from the genes. These 3 genes will code for proteins that allow the transport of lactose into the cell and for lactose to be broken down into glucose plus galactose. One of the genes codes for the enzyme beta-galactosidase, or what we call lactase. Okay. The operator is the region that codes for or allows the expression of the genes the genes to be expressed if the repressor's present or not. And the repressor binds to the operator region and prevents this gene from being transcribed. The repressor protein blocks RNA polymerase to prevent transcription of the genes, of these 3 downstream genes. So as long as this repressor is present, RNA polymerase cannot bind and cannot make these three enzymes or proteins. Now, if lactose is present, lactose naturally can break down and can be broken down into different products. But one form of lactose is called allolactose. It's a rearrangement of the lactose molecule. And we call allolactose the inducer. So, an inducer, here, let me change the color of the inducer. Okay. An inducer, in this instance, is allolactose, or we'll just say lactose. Okay. It's a form of lactose. The details, the biochemistry of which we're not prepared for, but nonetheless, the inducer is a substance that can bind the repressor and remove it from the operator. And in essence, that's going to allow RNA polymerase to transcribe the gene. So it induces transcription of the gene of interest. In the absence of the inducer, the repressor is bound to the operator region and overlaps the promoter, so RNA polymerase can't bind. I get no gene transcription. But the minute I start to introduce the inducer, the minute that lactose becomes available, lactose can bind to the repressor protein, and the repressor protein will have a higher affinity for the lactose than it will for the operator. And when those two come together, what happens is lactose will cause the repressor protein to change its shape. And as it does that, I'm going to read some of these definitions. And you need to look up the actual definitions of inducer and promoter and operator. But in essence, when lactose is present and binds the repressor, as it removes the repressor from the operator region, now RNA polymerase can actually bind here, and it will transcribe, and I'll get my mRNAs. Okay, it's very messy handwriting, pardon me, but I hope you guys know these videos are very crudely done because we're in a very short time crunch to get these videos up and loaded for due to COVID and having to teach online, and I absolutely hate it, but it's making me learn some new stuff. And I hope to, in the near future, reshoot all my videos and make them much nicer and neater and better done, more professional. But right now it's the cell phone, me, and this board with no edits, so bear with me. So, in the presence of the inducer, this one, in this instance, it happens to be lactose. Lactose will bind to the repressor protein, making it change its shape so that it can no longer bind to the promoter region. You have to remember, proteins start off as a straight chain, and then they fold up into a three-dimensional shape, which allows them to bind to something. But the reason that three-dimensional shape is what it is that there are internal hydrogen bonds and chemical bonds that cause the protein to fold up. If I break some of these bonds in here, the protein would start to unfold or change its shape. And so if I introduce another molecule here that binds up some of these amino acids, now this protein might change its shape completely and look different than when this inducer is available. So the repressor protein is expressed specifically to repress transcription of this gene because we don't need to waste energy and resources making it. But when the inducer comes in and binds and sticks to the repressor changes its three-dimensional shape and can no longer bind to the DNA. As it is released from the operator region in the presence of the inducer, RNA polymerase binds to the promoter and the operator and runs down the genes and transcribes them, and then those will be translated into proteins, and then those proteins will allow the lactose to be more lactose to be transported into the cell and broken down for energy. When all of the lactose has been broken down, the repressor can now go back to its original shape and bind back to the operator region, shutting the gene off. This is called an inducible operon. It's called an inducible operon because, and the definition of inducible operons are operons which are off, they're not transcribing proteins, unless they are turned on by an inducer, hence the term inducible operon. In the absence of lactose, then the repressor is bound to the operator and the promoter and prevents RNA polymerase from transcribing the gene. When the repressor is introduced, it binds to the, when the inducer is available, it binds to the repressor protein, changing its shape, opening up the operator and promoter region for RNA polymerase to bind and transcribe the genes. So an inducible operon is one that is off unless it's turned on by the presence of some inducer. And that's kind of how it works. Now the exact opposite of this is called a repressible operon. So some genes are on all the time, and then we can turn them off in certain conditions. So we're going to have basically the same setup, okay? But we're going to talk about what's called the tryptophan operon. One of the 20 amino acids that can be one of the 20 amino acids that is used to build proteins is called tryptophan. And tryptophan is also the stuff that people say is in turkey that causes you to want to fall asleep after you have a big Thanksgiving meal. But nonetheless, tryptophan is an amino acid. And when tryptophan becomes too abundant in the cell, we can turn off a series of genes using it. So in this instance, RNA polymerase is going to be bound here, and it's going to transcribe a whole bunch of genes. There's actually, I think, 5 genes in the tryptophan operon. We're not going to, the number doesn't matter to me, but the concept does. And so this one is turned on and transcribing or expressing the genes because RNA polymerase can bind to the promoter, run downstream. And when tryptophan becomes too abundant, there is a molecule in here called a repressor. And the repressor has a certain shape that does not allow it to bind to the operator. But when tryptophan comes and binds to the repressor protein, then this little complex can go bind to the operator region and prevent RNA polymerase from transcribing the genes. So, in this whole instance, it's the exact opposite of an inducible operon, and so these are called repressor operons. In a repressor operon, the gene is on, making the proteins. RNA polymerase can bind and transcribe the gene, they can then therefore go be translated. But in the presence of some chemical, in this instance tryptophan, when it binds to the repressor protein, the repressor then can fit onto the operator and block transcription of this piece of DNA. So, this is one simple example of the control of gene expression, how we can turn genes on and off. This one's used a lot for teaching this concept because it's present in bacteria like E. coli and many other bacteria. And so it provides us a sort of a little bit of insight into the manner in which genes can be turned on and off so that we can control their transcription and translation. Okay. I think that's about all the detail I want to go into. Let me check my notes one last time to make sure there's nothing that I've talked about or I've skipped. No, that's about it. So, a few quick key concepts I want to cover. The expression of genes is expensive in the terms of energy and resources like amino acids. There are many, many, many genes. Again, E. coli has over 4,400, I think it's 4,406 genes, I think, or something like that. 4,401 genes on the single circular chromosome. Not all genes can be transcribed all the time or the cell would run out of resources. Most of the genes, or some of the genes, are housekeeping enzymes that are produced 24/7. But other ones, like the enzymes for the breakdown of lactose, are not turned on unless they are needed, and that requires the presence of an inducer like lactose or allolactose. And that allows us to control gene expression, turn genes on, or in the case of tryptophan, we can turn a gene off when we no longer need it, okay? So the idea of control of gene expression is an important one because it allows us some insight and maybe some biochemical controls for us to control turning genes on and off at will if we wanted a particular protein to be expressed in a bacterium. For example, if I need a lot of beta-galactosidase, I can grow E. coli in the presence of lactose, and they'll produce a lot of this enzyme, and we can actually siphon off and isolate the enzyme and have some isolated beta-galactosidase around if we want. Then we could use that in certain media for the breakdown of lactose or things. So it allows for some applications for us to control the expression of genes if we want to. Anyway, listen, I hope you learned something. I hope you had as much fun as I did. Thanks for watching, and I hope to see you in the next video. If you learned something from this, please hit like and let me know. Thanks.